Good morning everyone. So, in the previous classes we have learnt about what are surface properties and bulk properties and how to characterize the surface properties and mechanical characterization of the materials. So, we have learnt about that. So, coming to the final characterization, which is biological characterization. So, this is the end game which determines whether your material is biocompatible or not, which is basically whether whatever the implant or devices you are producing, which will be taken by the host system or will be rejected by the host system. So, in biological characterization there are three major categories. One is in vitro biomaterial characterization and another is in vivo biomaterial characterization, and apart from this there is a separate section which is bacteria biomaterial interaction. So, in vitro is where you will do all the biological related experiments on a lab scale environment. In vivo where you will experiment the material in animal models like rat, pigs, canine species and all. So, bacterial interaction is mainly needed when you are looking for infection on the material surfaces and all. So, when you are implanting a material into the body, so within a nanoseconds the proteins will get adsorbed onto the material surface. So, based on the protein adsorption it determines how the cells are adhering onto the material surface, which will lead to the host material host cells and material interaction. If the cell attachment it continues, and if there is a proper cell attachment it will lead to spreading of the cells, and then which will lead to the differentiation and migration. So, all these steps will lead to complete integration of a material into the body. So, based on the application you have to decide what type of interaction you need. Like if you need a interaction between the cells and the material for the applications such as orthopedic implants, wound dressing and all. So, you need your material to be integrated with the host cell. But if you are looking into cardiovascular devices and all, where you do not need any cells to be adhered onto the surfaces and all, you should avoid the cell attachment on the material surfaces and all. So, coming into what are the aspects you have to look for the material to be a biocompatible. So, there are lot of aspects based on again application. So, until now whatever the surface property and the bulk property you have analyzed or characterized, those can be changed based on the results you are getting. But for this biocompatibility it is entirely dependent on the how the material is interacting with the cells. So, what type of cells you are using and what type of experiment you are doing, based on that it will interact with your material. Okay. So, whatever material you have characterized and made suitable for your application and all, it should be attached to the cells based on how effective it can be integrated into the system. So, coming into aspects, so first is sensitization, irritation and intracutaneous reactivity. So, this is mainly applicable for wound dressing materials. So, what happens is when you are putting a wound dressing on a particular wound, it should not cause irritation or it should not produce any allergic reaction on the surfaces and all. So, we have to test for that. So, that is that aspect. Then systemic toxicity. So, systemic toxicity involves what duration of toxicity you are going to observe over the period of time. So, toxicity, which is a commonly used term for studying the biocompatibility of any scaffolds, hydrogels or the materials. So, what you are basically going to do is whether the material you have produced is leaching out any compound or it is interacting with the cells and killing the cells. So, we have to check that using systemic toxicity. Genotoxicity, which is the material which can cause damage to the genes, so which can lead to mutation and all. So, implantation mainly observed when you are using a material which can affect the nearby tissues and organs also. If you are using orthopedic implants and all, it can have an effect on nearby bones and nearby tissues, muscular tissues also. So, based on the where you are implanting, you have to decide, you have to find out the factors affecting that. Hemocompatibility, it is primarily involved for cardiovascular applications where you need to your material to be fully blood compatible. And carcinogenicity, it is again linked to genotoxicity or the mutagenicity, where if there is a mutation, it can lead to cancer causing effects and all, which will lead to further rejection of your implants. Reproduction and development, it is not primarily used for all the materials, but if there is a possibility, if there is a possibility that your material leachate or your nanoparticles or the molecules which you are using can affect the reproduction system or the embryonic development stage, it has to be checked for that. Biodegradation, it is used for a lot of applications such as vascular sutures and staples, where you are using that material and it should be degraded over the period of time. So, you have to check those things and all, how the cells will degrade those materials and all, you will check in that biodegradation and all. Immune response is basically involving the total immune system, whether the introduced material is having any antigenic effect, so that it will trigger the immunogenic cascade, so that it will lead to inflammation of the site of implant. So, if you are introducing metal orthopedic implants and if it is causing leach out immunogenic responses and all, that place will have a inflammation occurring. So, these are the major aspects. So, in this one, commonly used test for assessing the biocompatibility or cytotoxicity, which is the initial test we have to do for all the materials, whether that material is killing the cells or it is having an inert effect or it is promoting the cell growth and all. Then hemocompatibility, where you have a blood related applications and all. Mutagenicity and carcinogenicity, where you will check for any change in mutation occurred in the system. Then bone remodeling, it is a separate topic, where you can estimate how much of the material is affecting the bone integration or bone growth and all. So, that is bone remodeling. So, in cytotoxicity, I will explain in detail all the assays. It has a direct and indirect contact, and couple of experiments, MTT assay and XTT assay, which is used for calculating the how much your material is toxic to the cells. Hemocompatibility, where these experiments will tell you whether your material is not causing any damage to the blood cells or it is leading to any platelet aggregation or the blood clot formation. So, mutagenicity and carcinogenicity, Ames test and chromosomal aberration and mouse lymphoma assay. So, in Ames test, we can do it in a bacterial study, where a bacteria, usually a Salmonella species will be there. So, in that one we will have a mutant which is having a histidine lack of histidine gene in that one. So, if there is no histidine supplied in the media, the bacteria cannot grow. Okay. So, you will introduce your compound. So, if it is causing a mutagenic reaction on the gene, it will convert the histidine and it will grow. So, that is the basics of Ames test. So, by this you can observe that whether the Ames test is whether the compound you are using is causing a mutagenic response or not. So, for all these tests, there would be a control where you will have a normal bacterial gene which has a histidine utilizing gene also. So, if it is not enhancing, your compound is not enhancing that, it can suggest that your compound is only changing the gene of the histidine lacking species. So, in chromosomal aberration, we will check how the chromosome is after the mitotic division and all. We will check with the compound and without compound. So, if there is a chromosomal aberration, there would be either insertion, deletion or translocation observed on the chromosomal structure. So, that can be observed in chromosomal aberration. So, mouse lymphoma assay is similar to Ames assay, but we will do it in a mouse lymphoma cells. So, in these cells which will have a thymidine kinase enzyme. So, that would be knocked out in a knocked out mice. So, if your compound is and they will supply a thymidine. So, if your compound can mutate the cells, then it can use that thymidine. So, by that you will understood that your compound is causing any mutagenic response or not. So, in bone remodeling, so the main factors we will look up all alkaline phosphatase assay. So, it in bone remodeling, when you are using a dental implant and or the orthopedic implants where you have a bone cements and all, that will lead to integration of the host cells, osteoblast cells and all, lead to formation of increased in calcium content, phosphate content, and osteocalcin is a major marker for bone cells. So, if there is an increase in these factors and all, you can actually estimate them using these assays and all, and found that whatever the bone scaffold or whatever your material is introducing, it has a effect on the improving the bone cell integration within your scaffold and all. So, systemic toxicity. So, LP enzyme, what it does is it converts phosphates to inorganic phosphates and all. So, other phosphate derivatives into inorganic phosphates. Those phosphates are major content of bone. So, they are the basic components, calcium phosphates. So, there are a lot of phosphates. So, based on that you will understood that if there is a higher activity, then there is a faster growth of bone tissue and all. Okay. So, systemic toxicity determines how long the implant you are going to use it in a system. So, acute system where you will use it less than 24 hours and all, which is people use catheters and all to do angioplast and other treatments, and subacute which is 24 hours to 28 days. Then subchronic 28 days to 90 days, urinary catheters and all, it would be within 2 months or 3 months they will remove it. Then chronic, which is above 90 days, so you have to test whether the material is not having any effect on the cells surrounding it. So, those chronic will involve all the bone implants and cardiovascular implants and all. So, based on this time duration, we have to check the material. It is exactly up till that time it is not having any adverse effect on the cells. And so, coming into toxicity, so this is a primary test we will do for all the materials, either it is hydrogel, scaffold or nanoparticles, whatever the material. So, we have to check whether it is accepted by the cells or not. So, that can be done based on 3 different methods: direct contact, indirect contact, elution or extract assay. So, all these variation is based on what is your sample and what type of sample you are having. Direct contact is where you have, as you can see in the figure, the first one where you have grown cells on the tissue culture plate and you put your sample on the top of it and check whether it is affecting any of the cells underneath it. So, that can be done for your nanoparticles or the not having too much load samples. So, if you are having too much load, it can destroy your cells automatically. So, indirect test, which is done using, there would be agar layer on the top of the cells and your sample would be there, in that which if your sample is leaching out some compounds or some aeroson or some other drugs are releasing from the material, then that can leach out and affect the cells underneath it. So, that is indirect contact. So, indirect contact can be done based on the how it is interacting with the cells also. So, as you can in the right hand side top figure, where first you grow the cells on a filter membrane. So, then after the cells are adhered onto the surfaces and all, then you invert it and then you will put the sample on the top. So, your sample is not exactly in contact with the cells, but the leachate of your sample would be going into the cells, and whether if it has any effect, it will kill the cells, or if it promotes the cells, it will have that effect. So, extraction or elution assays are used for soft materials where you can keep that material in a media for 24 hours or 48 hours based on your application. Then you have to extract the media. If there is a leach out from these compounds, you can take that media and culture it in that with the cells or with the indirect test method. So, you have the cells, you have the extracted media, that whether that leachate has any effect and all, you can test it. So, all these things are based on the sample size and how effective the leachate is coming out of the sample and all. So, if there is no leachate coming out of the sample, it will not have any effect and all. So, for that you can move on to the next one of test, which involves cell adhesion and proliferation. So, all these experiments can be done for those assays also, where you keep your sample at first, then you see the cells on the top of them. So, at this toxicity measures how much of your material is toxic and all. So, if you found out that material is not toxic, not causing any effect for all these assays and all, then you can go for cell adhesion. So, whether your material is promoting cell adhesion and all, because cell adhesion is very important when you are integrating your material with the host system and all. So, for that you will have the sample at the bottom and the cells would be seeded at the top of the material, and if over the period of time, if the cells are having adhesion property based on your material surface and all, that can be estimated also. So, for all the assays while involving biological characterization, control is very important. You should have a positive control and negative control. Negative control as such, there would be a standard of material. So, if you are testing a orthopedic implants and all, there would be a standard material, stainless steel would be there. So, you have to test that material for cytotoxicity. So, the negative control will not have any effect on the cells and all. There will be a positive control also. So, the positive control will have a maximum effect, maximum killing effect on the cells and all. So, based on those only you can, within that range only you can decide whether your material is cytotoxic or not. So, usually according to literature, if your material causes below 70 percentage decrease in cell viability, then that your material is not suitable for the biological application and all. So, now we saw how the experiment to identify your material is toxic or not. So, how exactly you identify that thing? So, that can be done by qualitative and quantitative. Qualitative involves you observe them visually using microscope. So, you can use a normal dye like Trypan blue or calcium AM and propylium iodide. So, calcium AM will go inside the cells and it will bind with the cytoskeleton and it will give a green fluorescence, and propylium iodide which will go and bind to the nucleus and it will give a red color. So, if there is a cell death and all, you can use these stains and identify whether the cells are live or dead. You can observe them under the fluorescence microscope and all. So, that is a qualitative test. And quantitative test, there are a lot of assays there. So, these are the commonly used one. So, MTT, XTT and MTS. So, all these are the tetrazolium salts. So, what it does is, so using a electron receptor, it will convert into formazole. So, the formazole will give a different color. In MTT, what happens is it will go inside the cells and mitochondrial dehydrogenase, it will reduce the MTT and it will form a formazole crystals. So, in MTT, it is a final step of the reaction where you will kill off all the cells and all, but in XTT, MTS and alamar blue and all, cells would be alive, because in MTT the formazole crystal formed is inside the cells. So, you have to estimate how much of the quantity of formazole has been formed and all. So, you have to rupture the cells and dissolve them using a solubilizing agent like DMSO and all to check the absorbance of the formazole crystals formed. Whereas, in XTT and MTS, which are water soluble tetrazolium salts, so it happens outside the cells itself with the help of phenazine methosulfate PMS. It involves plasma membrane electron transport, which converts these tetrazolium salts into formazole dye, and without rupturing the cells you can directly dissolve it in a culture media or the buffer and check the formation, amount of formation crystals formed and all.
The formation formed is not insoluble.
In MTT.
No, in XTT and MTS.
No, it is water soluble.
So can you use the same cells again for some other experiment?
Yeah, you can wash those cells and you can observe it for. But if you keep on repeating that, the cells will obviously die. So you can immediately take out and you can use that. So next to that is Alamar blue assay, which involves recessive dye. So this dye involves cytoplasmic redox reaction. So it goes into the cells and due to redox reaction it will convert into rhizophin, which will give a pink color. So compared to these above three methods, this is a highly sensitive method where you can estimate even a very small amount of cells also. And it has another advantage that it has a fluorescent property also. So you can using fluorescent spectrophotometer you can calculate even a minor change in the cell quantity also. So for all these things there would be a control. So where you do not treat your cells with any of your samples and all, and there would be another control where there will not be any cells, but whatever the material you are using will be in the media also, because those controls are very important, because all these involve colorimetric assays. So if your material is changing the media pH or any other adding colorimetric value to the system, then the results obtained can be of false positive. So to avoid that we should always have the control in all of the assays and all. So these are the methods for cytotoxicity. So after cytotoxicity, if your material is not toxic, you can go for the cell adhesion and all. So cell adhesion, it's involved the same list of procedures itself, where you can put your sample underneath the cells before seeding. So in cytotoxicity what you do is you seed the cells, you grow them, you make them for all these animal cell lines. It has to have a substrate to attach onto the attachment should be there. So you have a tissue culture plate, your cells would be properly spread on the tissue culture plate, and then you put on your material. If it is causing toxicity, that would be observed. In cell viability or the cell adhesion assay, what you do is you put your material and then you seed the cells onto the material surface. Material means any flat surface material or scaffold and all. So if based on your surface properties, whether it is hydrophilic or it has any receptor ligand for the cell receptors and all, based on that the cells will attach and you can estimate the amount of cells attached onto the material you are having. So coming into cell motility and cell migration assay. So these are next stage of characterization, where it is predominantly used in wound healing application. So in wound healing application what you have is there would be a wound separating two different tissues. So you same tissue, so you have to join them. So that can be done using this scratch assay. So you have a material, you seed the cells onto the material and you scratch them, and whatever your compound which will enhance the healing property and all, so you put that compound and check whether how the wound is getting closed. So as you can see in the figure, so the left side one is control and the right side one is treated samples. So they first grown cultures, then they using a small tip or spatula they create the wound. So in the left hand side there will not be any treatment at all. In the right hand side they have used Annona squamosa extract, which is a extract of custard apple leaves. So they have used it, and as you can see the wound has been closed within 2 days and all. So it has a wound healing property. So by this you can identify how much of how fast your wound can be healed using a different concentration of compound and different composition of the compound and all.
Like what were those HDF?
So HDF is human dermal fibroblast. So it is a skin cells. So dermal fibroblast. So whatever the material you are using, if it is non-toxic to one type of cell, doesn't mean it is non-toxic to all type of cells. So, each cell has a different property and different mechanism. So, based on the application, if you are going for the bone application and all, you have to check with the osteoblast cells, and if you are going for the vascular application, you have to check with the endothelial cells, and if you are going for the wound healing application, you have to check with fibroblast cells and all. So, each cells has a different attachment property and different effect on the material surfaces and all. So, that you have to always think. So, another important segment in biocompatibility is hemocompatibility. So, hemocompatibility, it is specific to blood related application. So, if a material is hemocompatible, it has a good blood compatibility. For example, if you consider heart valves, vascular grafts and all, there you have a continuous contact with the blood throughout the time. So, at the time of implantation and at the time of removal. So, there would be a continuous contact of the blood cells with the material. So, if there is any thrombus, so what is thrombus formation? So, thrombus is a blood clot, so which is formed by adhesion of platelets and fibrin mesh formed onto the surfaces. So, first platelet will adhere onto the material surface, then aggregation happens. So, if one platelet get adheres, it will send out extracellular signals which will activate the surrounding platelets and all. It will act as a homing beacon for making the other platelets to come into its nearby area, and it will form an aggregate, that is called platelet aggregation. So, when that stable platelet aggregate has been formed, there would be a parallel reaction coagulation cascade happens, which forms a fibrin mesh. So, that fibrin mesh acts as a network which will trap these platelet aggregates and form a blood clot. So, if you are having a vascular graft and if a blood clot is formed onto the on its surfaces and all, it will lead to stroke and other complications and all. So, in the right hand side, it is an example of how surface modification can improve the hemocompatibility. So, the top one, they have used CTI coated catheter. So, CTI is a chondrotrypsin inhibitor, so which will inhibit the clot formation, and bottom one is without any coating. So, as you can see, based on this study, you can identify a material can be avoid clot formation or not. So, what are the experiments you can do for hemocompatibility? The main experiment is platelet adhesion, because that is the initial step of thrombus formation. So, that can be observed using SEM. So, you incubate your sample with platelets, you extract healthy platelets from human volunteers, then you can check how the platelets is adhering onto the material surface, and you can observe it under SEM, or you can quantitative using previous whatever the cytotoxicity experiments. You can MTT assay also you can quantify. There is an assay called lactate dehydrogenase activity assay, which all involves the live platelets using the reductase enzyme. It will using a reductase enzyme and forming a colorimetric product, which will lead to quantification of these platelets. So, all these things you should have a standard. So, you have to count the number of cells and what is the number of cells having that effect, like in MTT for the particular absorbance, what is the number of cells. So, you have to make a standard and all. So, from that only you can calculate the actual number of cells on your material surface, or how much of has been reduced or increased from the control and all. So, standard is very important for all this biological. Other than platelet adhesion, there is a commonly used assay which is called hemolysis. So, hemolysis, as the name suggests, the lysis of the hemoglobin erythrocytes. So, whenever your RBCs are broken down, it will release hemoglobin that can be estimated using UV visible spectroscopy. So, based on the standard International Organization for Standardization, hemolysis index should be below 2 for the non-hemolytic material. It should not have any clot formation on the material surfaces and all. If it is slightly hemolytic, it is the range is 2 to 5, and if it is highly hemolytic, it is above 5, which is not at all recommended for any applications. So, hemolysis is calculated based on absorbance of the sample minus absorbance of the negative control by absorbance of positive control minus absorbance of negative control. So, what are the negative and positive controls are? Negative control is a normal saline solution which does not have any effect on the RBCs. Positive control can be either water or Triton X, where it will lyse the cells immediately on contact with the RBCs, and sample is whatever sample you are having. So, whether that sample will have any effect on RBCs, that is the sample. So, based on this you will find out the hemolytic index and identify a material is blood compatible or not. So, other than these methods, there are the partial thromboplastin time which is to estimate the amount of estimate the time required for the thrombus formation. So, you have your control material and you have your sample material, and in control material you put on your blood sample in both of them, and you add a coagulation factor in both of them. So, coagulation factor which will lead to thrombus formation. So, if your material is which is avoiding thrombus formation, there would be a delay in thrombus formation, whereas in this reference material it would be immediately formed. So, based on that you find out the partial thromboplastin time, and you can look into markers of coagulation cascade like C38, C58. So, all these are, there is a series of pathway involved for involved in the coagulation cascade. So, you can check for the particular markers whether it is in inactivated or activated state and all. From that you can identify that the material which you are inserting is activating the blood cells and causing this coagulation cascade and all, because it is a serious issue that if a small clot also forms, if it get detached from the surfaces, it can go and block somewhere in the arteries which is called emboli, that movable clot is called emboli. So, that can block the arteries and it can lead to serious issues. So, when you are looking for a blood contacting application, you should always do the hemocompatible studies and all. So, coming into bacterial interaction with the biomaterials, one of the major complication immediately arises when you are introducing a material into the system is infection. So, if there is an infection, there would be an immediate rejection, because if there is a bacterial infection on the material surface, it can lead to immune responses and all other toxic to the cells and also it would be immediately rejected by the cells. So, we have to make the material as much as possible to avoid any bacterial adhesion and forming a bacterial adhesion on the surfaces and all. So, this is majorly observed, and it is majorly observed in dental plaques, where you have a dental implants where you have lot amount of bacteria exposed to the material and all, and infection on urinary catheters. So, urinary catheters there would be a lot of bacteria excreted and all. So, there is another place, and orthopedic prosthesis there also, if there is a little bit of bacterial interaction is there, that can lead to infection, serious issues. In normal condition, the bacterial would be as a planktonic state which is suspended in the liquid solution, but when this bacteria is adhering onto the material surface, it becomes a sessile state which will lead to the formation of biofilm. So, what are biofilm? So, as a normal bacterial adhesion, it is reversible. So, due to shear force and all, it can be easily removed off from the surfaces and all. But when a bacteria adheres and it forms aggregates and all, it then changes its metabolism and secretes exopolysaccharides. So, these exopolysaccharides, what they do is they cover these bacterial aggregates and form a thin biofilm and all. So, these exopolysaccharides will protect them from the immunogenic cells and all, thereby it will be there for over a long period of time, and whenever the host immune system is low, these can come out of the exopolysaccharides and it can infect the nearby tissues and cells and all. So, biofilm is a major problem and your material should be avoiding the biofilm formation as much as possible. For biofilm estimation, you can there are two basic methods. One is turbidimetry measurement and another one is a plate count measurement. So, in turbidimetry measurement, you measure the number of bacterial cells based on the turbidity observed at OD of 600 nanometers. It is a standard for observing bacteria. You can estimate the amount of bacteria. So, you have a control, you have your sample and all. You inoculate the control with the bacteria and you have your sample and inoculate with the bacteria, and after the particular amount of time when the bacteria reaches its log phase and all, you will check for how much of the bacteria has lead to adhered onto the material surface and all. So, from that we have to wash out the bacteria adhered onto the surfaces and all and estimate using UV visible spectrophotometer.
Biofilm formation is caused only by gram-negative bacteria because only they have the lipopolysaccharide.
It's not exactly by gram-negative bacteria. It depends on the material properties also. Predominantly, it is on gram-negative bacteria. But if your material is having very rugged surfaces and all, bacteria can go and bind into the pits and wherever the cones are there. So, it can bind into the depth of the material surface and it would be stay there for a longer period of time. So, when it leads to aggregation, then it forms a biofilm. Both gram negative and gram positive different.
They don't study interaction of fungi with biofilm.
Fungi infection, it's not very widely studied because you need a like bacterial infection, fungi infection, you need a environmental condition, a different environmental condition than a bacterial condition. So, and it can be easily killed off fungi and all. So, bacteria, even if you sterilize it, there can it can form as a cyst and be available for a longer period of time. Even they study the sterilization techniques for the materials and all. So, after sterilization, how much of sterilization residual is still there, that also has to be studied according to the standards and all. So, bacteria is the major problem rather than fungi. So, coming into plate count method, where you estimate the amount of bacteria adhered using a colony forming unit. So, the procedure involves similar to turbidometry measurement. In plate count method, so you inoculate your sample with the bacteria and you take out the sample and you wash off the biofilm using solubilizing solution and all. Then you dilute those samples and you spread it on a agar plate. So, based on how many colonies formed, so each single cell will have form a single colony. So, based on how many colonies it has been formed, you can estimate the amount of bacteria adhered onto the material surface and all. So, the optimum plate count should be around 30 to 300. So, as you can see in the left hand side, it is a very dense one where it is not an accurate method, and in the right hand side only below 10 below 30 is there. So, that is also not standard one. So, it should be in the mid range between 30 to 300. So, that will give you the correct value of bacteria adhered onto the material surfaces. So, for all these things you have to characterize your materials before that surface characterization and bulk characterization. So, that you should have the idea that it should avoid the bacterial adhesion, it should promote the cell adhesion, it should avoid platelet adhesion. So, all those things you have to do before the biological characterization. So, how you can overall flow chart for how you can plan your plan for the producing a material is, first you have to categorize what type of device, either the orthopedic implant or you are going to be a wound dressing material or hydrogels or the nanoparticles. So, based on the application, there are a lot of standard description given already on ISO, ASTM FDA. So all those people, they have a standard protocols and all. You have to do all those tests for that. Then based on the relevant literature, you have to identify whether that will have the effect based for your particular application and all. Then you have to what are the testing condition and instrumentation needed. Then the sample size. So, based on the sample size also the cytotoxicity varies. So, there is a certain guidelines given by ISO that based on if you are using a nanoparticle ratio, how much of amount of nanoparticles you can use for the how much number of cells and all available. Because when you are taking it to in vivo studies and all, you will consider that for body mass ratio. So, if you are taking a capsule or drug, you will that would be based on how much body weight you have to take that drug and all. So, sample size occurs important for materials also. So, if your material is having toxicity, if you have a bigger sample and all, you will not use that much big sample when you come to application. So, you have to reduce the sample size and all. Then you have to finally go for cost and time involved for developing that material. So, these are the standards given by ISO. So, each of the standards has a different set of guidelines for performing different applications such as 109935, which only involves in vitro cytotoxicity. Whatever the toxicity experiments I have mentioned, it will give you the clear guidelines of whether if your material is flat surface and all, how you can do, and if your material is scaffold or hydrogel and all, how you can do. So, like that each of these standards will give you what are the parameters you have to follow for the characterizing the biological compounds. So, if all this characterization has been done, then you have to finally take into in vivo. So, in vivo also similar to in vivo in vitro only, but after implanting into a animal model, you have to sacrifice the animal and take out the sample again out and check for the histopathology and all surrounding the implanted devices and all. So, all those things you have to again how much of cells are added onto the surfaces and all, whether it has caused any immunogenic responses and all. So, all those things you have to do for that.